Hi everybody, in this series of videos we want to talk about different imaging techniques which are used in the field of biology and nanomedicine. We start with the fluorescence microscopy, which uses the fluorophores. The fluorophores are excited by light at some specific wavelengths, and then in response they will emit lights at the longer wavelengths with the lower energy. But before getting into the details of that, let's have a look at the penetration depth of different lights within the tissue. As you see here, the blue light can penetrate into the body for about 1 millimeter, while the red light can penetrate for about 4 millimeters. And why is this happening? This is due to the different absorption coefficients of two molecules which are found within our body. One as the hemoglobin and one as the melanin. Okay, what is the hemoglobin? Hemoglobins are the oxygen transfer agents within our red blood cells. The oxygens will be absorbed onto the surface of the hemoglobins, and through the bloodstream they will deliver these oxygens into the cells. On the other hand, we have the melanins. The melanins are responsible for the color of the skin and the color of your hair, and they are produced by the cells which are called melanocytes. Okay, as you know, the red blood cells are ubiquitously found within the blood, and the melanocytes are found on the skin of the tissues. And because these materials have high absorption coefficients, they will absorb the light. As you see here, this curve related to the absorption coefficient of the hemoglobin, and this curve for the melanin. The coefficient related to the lights which have shorter wavelength, the blue, is higher compared to the light at longer wavelength, the red. So here in this region, because the absorption coefficient is larger, the light will be absorbed, and as a matter of fact it cannot penetrate into the tissue. How about in this region, for the longer wavelengths, the absorption coefficient is smaller compared to the blue light, and this will lead to higher penetration depth for the red light and shorter penetration depth for the blue light.